Good day, everyone. This is Giselle Farah Abelia, along with Critalin Datan and Roshime Joloya. We are the Group 3, and we are here to present our research proposal in partial fulfillment for the course subject Developmental Biology 3. Our research proposal is entitled Toxicology Study of the Nanomaterial AGNP or Silver Nanoparticle and its effects on the survival rate of Drosophila melanogaster. Over the years, the nanotechnology industry has been rapidly growing due to its physico-chemical properties of the new produced nanomaterials. It has gained a public interest because of the needs and applications of nanomaterials in many areas of human endeavors, such as agriculture, industry, medicine, and public health. The widespread expansion of this market indicates the increasing presence of nanomaterials in the environment. In our study, we will be focusing on nanotoxicology. Nanotoxicology is an emerging discipline and there are only few studies have been performed up to the present. The nanotoxicology is referred as the science that investigates the mechanisms of interaction between nanoparticles and humans. In the study, the nanotoxicity of silver nanoparticle will be studied. It is because silver nanoparticles represents one of the most prevalent nanomaterials in commercial products and biomedical applications. Silver nanoparticles have become the fastest growing product category in nanotechnology due to their thermoelectrical conductivity, catalytic activity, and nonlinear optical behavior. Each year, it is estimated that approximately 320 tons of silver nanoparticles are produced and used worldwide. According to Vance et al. 2015, there were over 438 products produced that were advertised with nanosilver, and because of the exceptional broad-spectrum bactericidal properties and biocompatibility of silver nanoparticles, the silver nanoparticles are widely applied in medicine, cosmetics, textiles, and other consumer products, including food containers and washing machines. Apparently, silver nanoparticles can cause ill effect on humans as well as the environment. There were numerous reports mentioning that nanosilver cannot differentiate between the microbes that are useful or harmful to the ecology. The free silver ions have harmful effects on living beings. Exposure to soluble silver compounds may produce toxic effects on liver and kidney and can cause irritations in the eyes, skin, respiratory, and intestinal tract, and it also brings untoward changes in the blood cells. With the evident reports on silver nanoparticles, these suggest that the negative impact of these nanoparticles on any living organisms are of major concern. And for the objectives, in order to monitor the impacts of silver nanoparticles on human health, our group, we plan to increase our knowledge of the potential toxicity risks associated with silver nanoparticles exposure, specifically to test if silver nanoparticles intake can affect the survival rate of Drosophila melanogaster through an in vivo model system.
The D. melanogaster is an intensely studied and powerful organism that was introduced 100 years ago by T.H. Morgan. It is characterized as a small and sexually dimorphic organism with females of about 2.5 millimeters long, while males are slightly smaller with darker backs. Its genome is comparably smaller than the typical mammalian genome. However, it is 60% homologous to the human genome. It is also accounted that the D. melanogaster has homologous genes to that of 75% of the genes that causes human diseases. Aside from its morphological and molecular characteristics, its developmental and behavioral aspects are closely similar to that of humans, which makes it a better subject for research studies. Furthermore, one of the most important characteristics of the D. melanogaster is its short life cycle that allows the rapid production of generations of of numerous progenies for studying. Drosophila melanogaster studies. The D. melanogaster has been proven to be an effective model organism for the study of genetics. In a study conducted by Ayer and his colleagues in 2018, the neurodevelopmental defects that result to autism was investigated through the 16p11.2 gene deletion in D. melanogaster. The researchers hypothesized that the pervasive genetic interaction of multiple dosage-sensitive genes is the one that causes the neurodevelopmental defects rather than being caused by a single causative gene. The researchers chose the model organism D. melanogaster in this study due to having a system that is sensitive to genetic perturbations but can still carry out the different interactions needed in the study without compromising the organism. In addition, the D. melanogaster was subjected into a series of quantitative and high-throughput assays. The main objective of the study was to give a systematic characterization of the phenotypes, function, cellular mechanisms, and interactions of conserved homologs of human genes 16p11.2. Aside from genetic studies, the D. melanogaster can also be used in studying host microbiota interactions. For example, in a study conducted by Trinder and his colleagues in 2018, they used the D. melanogaster as a model organism for high-throughput host microbiota interactions instead of using germ-free mouse. They chose the D. melanogaster due to its affordability, convenience, and replicability. Moreover, the simplicity of its microbiota was also considered. It has a low microbial diversity of about 1 to 30 species and is mostly dominated by the organisms Lactobacillus and Acetobacter. It is further stated in the study that the simplicity of this microbiota structure can simplify the complexity in understanding the effects of a microbial species that was introduced to a greater community and its hosts. Silver nanoparticles.
So for the research methodology, first we have obtaining Drosophila and culture. So 5 bottles containing 100 fruit flies will be collected at the Carbon Public Market, Cebu City, Philippines. So we have chosen this site for the reason that these flies can be frequently seen in rotting fruits, decaying vegetables, and some plant matter. And this place is perfectly fit for the description where we can see samples of Drosophila melanogasters. Then, the collected flies will now be cultured for 24 hours, supplemented with the synthetic medium as food. This synthetic medium includes the ingredients such as water, agar-agar, banana pulp, and a small amount of yeast. This medium was proven by Pearl is a good culture media for Drosophila. After being enclosed for 24 hours, mating is expected to happen and larvae are also expected. So, we will be collecting 600 larvae from the 5 bottles and we will transfer it into new vials. So there should be 60 vials. 10 vials will be labeled for the control group and the other 50 for the different um, experimental group. So the first 10 vials is for the 6 hours exposure to silver nanoparticles, another 10 vials for 12 hours exposure to silver nanoparticles, and another 10 vials for 18 hours exposure to silver nanoparticles, another 10 for 24 hours exposure to silver nanoparticles, and another 10 vials for 48-hour exposure to silver nanoparticles. And each vial will contain 10 larvae. So next, we have exposure of silver nanoparticles to Drosophila through food. This is to analyze the effects of silver nanoparticles to the survival rate of Drosophila melanogaster when exposed to silver nanoparticles for different length of hours from the larval stage. Each vial will be labeled with various time length, will be cultured in the synthetic medium suspended with 80 mg per liter dose of silver nanoparticles. Additionally, for the control vials, larvae will only be cultured in the synthetic medium without suspension of silver nanoparticles. The Drosophila in the vials with various time lengths such as 6 hours, 12 hours, 18 hours, 24 hours, and 48 hours will then be transferred to a new vial after the length time of exposure respectively. After exposing the larvae into different time lengths, it will be transferred into the new vials with only the synthetic medium. The survival of these flies will now be observed every day until the fly's end of life. So for the survival assessment of the Drosophila, both the control group and the group with treated with silver nanoparticles flies will be observed according to the survival rate. The survival rate will be determined by counting the number of flies that will grow into an adult fly in each vial. So for the statistical analysis, it will be performed using the t-test. T-test will be used to compare the control group, which is flies cultured under the synthetic medium, and the experimental group, which is the flies cultured in the synthetic medium, with silver nanoparticles exposed in 5 various time lengths. It is to determine if the treatment has an actual effect on the survival rate of the flies by comparing it to the control group. So that would be all. Thank you.